Hey everybody, we're going to start talking about DNA again, and specifically we're going to start by talking about how DNA is compressed and packaged together into these structures called chromosomes that we find within the cells of the body, or the nuclei of the body. But before we do that, what I'd like you to do is go back and review the structure of DNA, this double-stranded DNA molecule. And some things I'd like you to be able to do are, one, to recognize the individual single strands of DNA and the individual nucleotides that are making up that single strand of DNA, as well as the covalent bonds that are holding those individual nucleotides together. And then another thing I'd like you to review and to be able to talk about is how these two individual single strands of DNA are held together, linked together to form a double-stranded DNA molecule. And that's going to involve something called complementary base pairing. And you're going to want to look at the base component of the individual nucleotides, and also these intermolecular forces, these hydrogen bonds that are linking those complementary bases together. Okay, so I have a couple questions for you. And the first one is, where do we find DNA in the body? And the answer to that is that we find DNA within the cells of the body. And more specifically, we find DNA within the nucleus of the cells. And even more specific than that, this DNA is packaged together into structures called chromosomes. And we find these chromosomes within the nucleus of the cells of the body. Next question. What does DNA do? Well, DNA contains genes. And genes contain the instructions to build different proteins. What are some examples of proteins in the body? And these are all proteins that we've talked about thus far. Enzymes, antibodies, hemoglobin molecules, hormones, muscle tissue. These are all examples of proteins. And it's important to realize or to remember that the body is constantly making new proteins as part of normal growth, development, and maintenance. And so we rely on this DNA to provide the instructions to build these proteins. Okay, so let's start talking about chromosomes, and let's start by looking at this figure and figuring out what's going on here. So here's our double-stranded DNA, and as we're going to find out, the double-stranded DNA is packaged together through all these different mechanisms to form these structures called chromosomes. If we were to take all the DNA within a nucleus and just string it out, there was no way it'd fit within the nucleus of a cell. Okay, it'd be much too long. And so we need to package it into these structures that we call chromosomes, and then that can fit within the nucleus of a cell. Now, with regards to chromosomes, what does the number 46 mean to you? Well, 46 is the number of chromosomes found within almost all the cells of the body. There are some exceptions. Sperm and eggs, as we'll talk about, have a different number of chromosomes. And then there's some cells like red blood cells that don't have a nucleus at all, so they don't have any chromosomes in it. But the majority of the cells in the body are going to contain 46 chromosomes. And that's what we're looking at here. These are all 46 of these individual chromosomes. So the first level of DNA compaction into chromosomes occurs when this linear DNA wraps around proteins called histone proteins And by doing so, forms these structures called nucleosomes. So each one of these is an individual nucleosome. And we can take a closer look at that up here. So we have this complex of histone proteins, and they all have different names and numbers and whatnot. But as you can see, the linear DNA is wrapping itself around these histone proteins and forming these structures called nucleosomes. So this entire structure here would be called a nucleosome. And a good analogy for this is a garden hose. You have this long garden hose, and yeah, you can leave it, you can leave it just laid out across the lawn. But if you want to compact it, if you want to make it nice and tidy, then you can roll it up on this wheel, and now you have a whole bunch of hose or a whole bunch of DNA into this one structure that we call a nucleosome. And then there's another number of levels of DNA compaction that occurs for DNA to fit within a chromosome. So here's that first one. First of all, here's DNA wrapping around the histone proteins to form a nucleosome. And then what happens is that these nucleosomes are going to fold together. And by doing so, they're compacting themselves. And then on top of that, the DNA loops back and forth. And then on top of that, it becomes compressed. And then on top of that, it becomes tightly coiled together. So you have all these different levels of compaction that occur, and the end result is a chromosome that contains a whole lot of linear DNA in it. Okay, it's a whole long string of linear DNA that we've packaged together to fit within the chromosome. And we need to do this because, again, if DNA wasn't compacted together, if it was just linear, there's no way that it would fit within the nucleus of the cell. And then one thing that I want to quickly point out, most of the DNA is found within chromosomes within the nucleus of the cell, but there is a small amount of DNA that is found within the mitochondria, and this is called mitochondrial DNA. And most of this DNA is going to contain genes that code for proteins that are involved in cellular respiration, for oxidative phosphorylation, for instance. So there is some DNA that's found within the mitochondria. And a couple interesting things about this is that, one, it's located on a circular chromosome. Okay, so not something that happens within the nucleus. And also, all of this mitochondrial DNA is inherited solely from the mother. So all of the cells of the body can be classified as either somatic cells or sex cells. And maybe the easiest way to think about this is that sex cells in females are the eggs, and the males are the sperm, and then any other cell besides that is considered a somatic cell. So skeletal muscle cells, nerve cells, skin cells, these are all somatic cells. And within these somatic cells, let's look at the skeletal muscle cell first. And skeletal muscle is actually multinucleated. They have several nuclei within a single skeletal muscle cell. And these are all the individual nuclei. Over here on the nerve cell, in the cell body, there's the nucleus there. And then here's the skin cell, and as you may be able to find or point out, that's the nucleus there. And how many chromosomes do we find within each of these nuclei in the somatic cells of the body? Well, you already know this. There's 46 chromosomes found within the nuclei of the somatic cells. And so the question then becomes, well, how many chromosomes are found in the sex cells? How many chromosomes are found in the egg and the sperm? And the sperm's telling you this here. Each of these sex cells has 23 chromosomes. And my question to you is, why is that the case? Why do sex cells, sperm and egg, only have 23 chromosomes? Well, this has to do with fertilization. Essentially, what's going to happen with fertilization is a sperm is going to fuse with an egg, and they're going to combine their chromosomes to form a new organism. And we want that new organism to have 46 chromosomes in it. So 23 are going to come from the mother, 23 are going to come from the father. And this is a better illustration of that. But here we have a sperm, and up here in the head of the sperm, there's 23 chromosomes. Here we have the egg or the ovum, and within the nucleus, there's 23 chromosomes. And what's going to happen is during fertilization, the sperm merges with the egg, and they're going to combine chromosomes to form a new single-celled organism, and that new single-celled organism is going to have 46 chromosomes in it. And this is maybe a better illustration of that over here. This is the egg with the nucleus. Here are the individual sperm. And one lucky little sperm is going to make its way through the egg into the cell. And what happens next is they're going to combine their chromosomes. They're going to mix their chromosomes. So 23 from the mother, 23 from the father, and now fertilization is complete, and you have a new single-celled organism that we call a zygote, and that zygote's going to have 46 chromosomes in it. What happens next? Well, this typically occurs in the fallopian tubes in the female reproductive tract. And after that zygote is formed, it immediately begins cell division. So it divides and becomes a two-celled organism. Then each of those divides and you get to the 4-cell stage, 8 cells, 16, et cetera, et cetera, et cetera. And what I want to point out is that each one of these individual cells that's forming in this new organism, each one of these cells has 46 chromosomes in it. So these are all somatic cells. And that this new organism is going to make its way down the fallopian tube, and then it's going to get to the embryonic stage where you have thousands and thousands of cells, and then it's going to implant in the uterus for development. But what I wanted to point out here is that after fertilization, you get cell division, and each of these new cells is going to have 46 chromosomes in it. And then something interesting, I was going to say decide whether to bring this up or not, but in the case of males won't start producing sex cells, sperm, until they reach puberty. But what's interesting about females is that females produce all of their eggs, or at least that we know of, in utero. So if this is a female, then at some point, and I don't know what stage during fetal or embryonic development this occurs, but she will start producing sex cells at that point. But again, I don't know when specifically that occurs. But again, the main overall point that I wanted to make here is that during fertilization, you combine sperm and eggs. So now you have 46 chromosomes, and that cell immediately starts dividing, and all of those subsequent cells are going to have 46 chromosomes in it, potentially with the exception of female sex cells, again, which are formed in utero. Okay, so moving on. Here we have this nice picture, and this picture is called a karyotype. And what this represents is this is a photographic representation of the chromosomes within a cell. And normally, the chromosomes are scattered throughout the nucleus. They're not neatly organized like they are here. But what they've done here is they've been stained, first of all, and they've been arranged in what we call pairs. So there's 23 pairs of chromosomes here. Here's the first pair, the second pair, etc., all the way down to the 23rd pair. And if there's 23 pairs of chromosomes, then how many total chromosomes are we looking at in this karyotype? We're looking at 46 chromosomes. So these can be the chromosomes from any of the somatic cells in the body. They all have 46 chromosomes. But again, what we've done is we've arranged them into what we call pairs. And we call each of these pairs, we call these homologous chromosomes or homologous pairs. So we have 23 sets of homologous chromosomes, which gives us a total of 46 chromosomes within each nucleus. Now, where are these chromosomes going to come from? Well, half of them, 23 of them, are going to come from your mother via the egg, and the other 23 are going to come from your father via the sperm. But there's something else going on here on top of that that's pretty cool. So for those 23 chromosomes from your mother, you may ask, well, which 23 chromosomes come from the mother and which 23 come from the father? Well, what happens is that within each homologous pair, so we'll say within each homologous chromosomes, one of those chromosomes is going to come from your father, and the other chromosome in that homologous pair is going to come from your mother. So within each homologous pair, within each homologous set of chromosomes, one of those is going to come from your mother, and the other one is going to come from your father. And something that I want to point out is that our entire genetic code, what we call our genome is located within these 46 chromosomes. Our entire genome is dispersed between these 46 chromosomes in each somatic cell of the body. So now let's take a closer look at one of these pairs, at one set of these homologous chromosomes, and maybe we'll just say this is set number 3. And so my first question for you is, how do these chromosomes within a homologous pair, how do they differ? And the answer to that, like we just covered, is that one of these chromosomes is going to come from your mother, and the other one is going to come from your father. And then the next question is, how are they similar? How are these chromosomes within a homologous pair, how are they similar? Well, the answer to this is that each of these chromosomes contains the same genes. And they not only contain the same genes, but these genes are located at the same location or locus or loci on each chromosome. So for instance, within homologous pair number 3 here, each of these chromosomes are going to contain the same genes at the same location. So for instance, maybe these chromosomes are both going to contain the gene for eye color. Maybe they're both going to contain the gene for freckles. The point is that whatever genes these homologous chromosomes contain, they each contain a version of that same gene. So another something interesting to point out is that within the entire human genome, within the entire genetic code of a human, there's about 21,000 what we call protein encoding genes, genes that contain the instructions to build proteins. And each homologous pair only has a portion of them. So if we look over at this karyotype, maybe homologous pair 1, maybe this only contains 1,000 of those protein encoding genes. And then pair number 4, maybe that contains 4,000 of those protein encoding genes. So each pair only contains a fraction of those, but collectively, all 21,000 of these protein encoding genes are located somewhere on these 46 chromosomes. So the entire human genome, all of the 21,000 or so protein encoding genes are found somewhere on these 46 chromosomes. And each homologous pair contains the same genes, but they only contain a portion of a fraction of those 21,000. The next thing I want to talk about is something called ploidy, or ploidy number. And this is represented by the lowercase letter n. And what ploidy refers to is the number of chromosomes in a homologous set. So looking at our karyotype here, how many chromosomes do we have in each homologous set? And the answer to that is 2. And so because of that, humans are considered diploid. And we write that as 2n. Again, because there's 2 chromosomes in each homologous set. And to be a little more complete, the way we write this out is 2n equals 46. 2n, again, means that within each homologous set, there's 2 chromosomes. And 46 is the total number of chromosomes in the cell. So we have 46 total chromosomes, and those chromosomes are arranged in sets with 2 chromosomes in each set. And all of the somatic cells in the human body are going to be 2n equals 46. Which begs the question, are there any cells in the body that are not diploid? And the answer is yes. There are some cells in the body that are called haploid. And what that means is that there's just 1 chromosome in each homologous set. And take a guess at which cells are not going to be diploid. Those are going to be the sex cells. And the sex cells only have 1 chromosome in each set. And so it's written as a single N. That's called haploid. And there's 23 total chromosomes in a sex cell. And so that's where the 23 comes from. And this is a good representation of that. So there's an egg, there's a sperm, and each one of these sex cells has 23 chromosomes within it. But those chromosomes, there's 23 sets of chromosomes, but within each set is only one chromosome, and that makes it haploid. So this egg is N equals 23. The same goes for this sperm. There are 23 chromosomes, but each of those chromosomes is in its own set. Okay, there's only one chromosome in each set. So again, that makes it haploid with a total chromosome number of 23. But what happens with fertilization? During fertilization, the sperm and egg are going to combine their genetic material. And so, bring this closer up. Half of the chromosomes are going to come from the mother. And at the same time, half of the chromosomes are going to come from the father. And the key here is that the mother and the father both contribute one chromosome in each homologous set. So in these sex cells, there's only one chromosome in each set, but then you combine the sperm and egg together, and now you have two chromosomes in each homologous set. And that's what takes us into this diploid state, going from a haploid to a diploid state. And just one more time, with these sex cells, there's 23 chromosomes, and all 23 homologous sets are represented, but again, there's only one chromosome in each set. Same goes with the sperm. And then during fertilization, what the mother and father each contribute is one chromosome to this homologous set. And so now we have two chromosomes in each homologous set, and that takes us to a diploid state. Now, there can be errors in the number of chromosomes that are found in one or more of these homologous sets. And this is called aneuploidy, is the generic term to describe this. It just means abnormal number of chromosomes. And probably the best known, most recognized example of this is when you have an extra chromosome in the 21st homologous set. And this results in a condition known as Down syndrome. So what causes this to happen? What causes an extra chromosome to be inserted in the 21st, for instance, or any other homologous set? And typically what happens is that this is an error during a process called meiosis. Meiosis just represents division of the sex cells. And the reason the sex cells divide is to achieve this haploid 23 chromosome state. And what happens with Down syndrome and other aneuploidies is that during this division, instead of the egg ending up with 23 chromosomes, it may end up with 24 chromosomes. And so it's already diploid for that 21st set. And so then what happens during fertilization, when you combine that genetic material, the offspring, the child, is left with an extra chromosome in that 21st pair, in that 21st set. And so it's an error during the process of meiosis. You can also have chromosomal abnormalities, aneuploidy, in the 23rd set of chromosomes, which are the sex chromosomes that are going to determine your genetic sex. And males have what we call an XY condition, and females have an XX condition. And again, there can be abnormalities here that result in different results. So in males, for instance, one of those conditions is if you have triploidy, and specifically you have an extra X chromosome. So you have an XXY condition, and this is known as Klinefelter syndrome. And these are genetic males, but because of this extra X chromosome, it can have some sexual repercussions. It can result in sexual immaturity, including a lack of sperm production, and it can also result in breast swelling. In females, so females have an XX sex chromosome, and if you get triploidy there with an extra X chromosome, then that can result in what's called triple X syndrome. And the consequences there can be menstrual irregularity. It can make them tall and thin. Or if you just have a single X chromosome, you're lacking the other X chromosome, then that can result in Turner syndrome. And again, that can have different repercussions. In this case, it can result in sexual underdevelopment and also a short stature and a web neck.